In this video lecture, we are going to be covering the topic of sex-linked inheritance. So we need to understand the chromosomes that are responsible for giving traits that are sex-linked. In humans, there are your set of sex chromosomes, which determine the biological sex that organism was born with. And the sex chromosomes are either XX representing a biological female and XY representing a biological male. And so this karyotype here is just showing the breakdown of human chromosomes. We have 23 pairs or 46 chromosomes in total. So pairs 1 through 22, those are your autosomes that give you just basic traits throughout your body. And then any traits related to the sex of the organism are on those sex chromosomes. So again, either XX representing a biological female and XY chromosomes representing a biological male. And the traits that we're going to be talking about are found on these sex chromosomes specifically. They're not found on any of the other pairs of chromosomes. And there are specific traits that are found on these chromosomes like hemophilia, color blindness, male pattern baldness. So when we talk about these traits, we also need to specifically talk about those sex chromosomes because these traits are found on them. So it's important to understand, are we talking about a female or a male, and what version of the trait do they have? And you'll notice that in terms of the sex chromosomes, so females have 2 X's, males only have 1. That Y chromosome that males have is much smaller in size, and there are a lot less traits on it because it's a lot smaller in size. So when we're talking about traits that are X-linked, that means we're talking about traits where the allele for the trait is found specifically on the X chromosome. It is not found on the Y chromosome. So because females have 2 X's, we're going to use exponents to represent the allele of the trait on that X chromosome. So if a female has 2 X chromosomes, she's going to have 2 alleles. So we're just using the letter B here to represent this trait for whatever we're tracking. So because she has 2 X's, 2 X chromosomes, she also has 2 of the B alleles. And in this case, because both B's are uppercase, that means this female would be dominant for the trait. Again, another example of a female X-linked trait would be, again, having 2 X chromosomes because this individual is a female. On the first X chromosome, they have a dominant allele, or the uppercase B. The second chromosome has the lowercase or recessive allele. So because the trait itself is big B, little b, overall this female will be dominant for whatever the trait is that we are tracking because that one dominant allele masks the recessive allele. The last female genotype is where both alleles for the trait are recessive. So we still have the 2 X chromosomes, but their exponents showing the trait are both little b, little b. So this female would be recessive for whatever trait it is that we are tracking. For males, there are only 2 different types of X-linked genotypes that we can see. Because this is an X-linked trait, that trait, the B trait here, is only found on the X chromosome. We only see the exponent next to the X. We do not see the exponent next to the Y because that Y represents the Y chromosome. This trait is not found on that chromosome at all, so we don't put anything next to the Y. So for this male, because the exponent is a dominant allele, or the uppercase B, they would show the dominant phenotype. This genotype down here at the very bottom is again a male because it's an XY sex genotype, but the exponent representing the B trait is recessive, so the male would show the recessive phenotype. So you do just need to get comfortable understanding that the X's and Y's represent the sex and the exponents represent the trait that we are tracking. And we only use the X and Y when the trait is specifically found on the sex chromosomes. This is only used for sex-linked traits. Occasionally, but very rarely, we'll talk about Y-linked traits. So these are traits only found on the Y chromosome. These would be specific genes that males have that females would not, because females do not have Y chromosomes. And because of that, their genotype for a Y-linked trait, there would be nothing here. They're just XX. So there's no exponents here for females. They just possibly cannot have that trait because they don't have a Y chromosome. For males, their sex genotype is XY, and this is Y-linked trait, so their X's would also have nothing attached to it, no exponent. The Y chromosome would have an exponent for whatever trait we're tracking. So in this case, if the exponent is an uppercase allele, that would mean the male is dominant for the trait. And if it is a lowercase allele, the male would show the recessive phenotype. So Y-linked traits, we don't really see that often. They're not very interesting because they're very specific to males only, and females do not have Y chromosomes. So one other piece of vocabulary that you need to know is the word carriers. And so what we mean by a carrier is that many of the traits that we are tracking that are sex-linked, in order to have the trait, you need to have both recessive alleles. So you need to have 2 copies of the lowercase allele. Females can be carriers, meaning that they're heterozygous for the genotype. So in this example here, we have a female. We have 2 X chromosomes, so we know it's genetically a female. The trait we're tracking is the color blindness versus normal colored vision. So big B would represent having full color vision. The little b allele represents being colorblind. So a female can be a carrier if she has one big B dominant allele and one recessive little b allele. So this female would have colored vision. She would not be colorblind, but she is carrying that colorblind allele. And we'll see why this is important in just a second. But it's important to note that when we're dealing with sex-linked traits and females are heterozygous, meaning they have the 2 different alleles, we would call them a carrier. And this is important for when we go to do Punnett squares. And we'll see that females that are carriers, they have the potential to create offspring that do have that recessive X-linked trait. So let's go through and see an example of a Punnett square problem done out so we can understand how these traits are passed on to offspring. So the first trait we're going to look at is hemophilia. It's an X-linked trait, so that means it's going to be attached to those X chromosomes. And it's recessive, meaning that to have hemophilia, you need to have the lowercase h or recessive allele. If you have the dominant allele or the uppercase H, you would not have this blood disorder, no hemophilia. So we're going to cross 2 individuals. So we have this female, and we know that because there are 2 X chromosomes. This female is big H, little h for hemophilia, which means this female is a carrier. She has the heterozygous genotype for hemophilia. That big H means she has the dominant phenotype, meaning she does not have hemophilia, but she is carrying that recessive allele for hemophilia. We're going to cross that female and with a male, and we know it's a male because it's the XY sex genotype. And this male has the dominant phenotype because there's only one X here, and it's an uppercase H attached to it. So this male would not have hemophilia as well. So both parents in this case do not have hemophilia. So let's create this Punnett square. And we're going to start by putting the female genotype across the top of the Punnett square where we split the 2 alleles. So we have the X with the big H and X with the little h. Then we'll put the father, the male genotype, down the side. So he is the X with the big H dominant allele and the Y that has no exponent attached because this trait is on the X chromosome. And now we'll just go in box by box, filling in the 2 genotypes from the top and the side. And that is my cat saying hello to everybody. All right, so this first box would be X big H, X big H. The next one would be X big H, X little h. We will have X big H with the Y, and then X little h and a big Y. So if we look at the phenotypes of these offspring, we'd see 50% of the offspring, there's a 50% chance that you get a female offspring that does not have hemophilia. One of these would be a fully dominant genotype, and one of these represents a heterozygous or carrier genotype. There's a 25% chance that the offspring would be male without having hemophilia, because one X would make this a individual without hemophilia. And then lastly, there is a 25% chance that the offspring from these two individuals would be male with hemophilia. And this male got that hemophilia allele from the mother who was a carrier. So the male did not have hemophilia whatsoever, but the mother, being the carrier, gave this potential offspring that recessive allele. So that is why we call the females carriers if they have the recessive allele but are showing the dominant trait, because they have the ability to pass that trait down to a potential offspring. So even though both parents do not have hemophilia, there is a chance that they would end up with a male offspring with hemophilia. So let's look at a second example here. So the next example of an X-linked trait, it's a common one. It is male pattern baldness. So this is where you see males losing hair on the top of the head, but they still kind of have hair along the sides and back of the head. And so what we're going to do is we want to cross a male that is bald with a female that is a carrier for baldness. And so for this trait of baldness, big B would be the dominant trait, representing a full head of hair. The little b would represent the male pattern baldness. So we need to create the genotype for the two parents. So we'll start with the female. So we know that she is a carrier, so we use the XX genotype. And because she's a carrier, she's got the big B and little b allele, meaning she has a full head of hair. The male genotype is XY, and on that X chromosome, we have the recessive allele for baldness, because we know that the male here is bald. And so now we will fill in box by box. So the first box, we have the two X's with the genotype for baldness as big B, little b. Then we have the XX genotype with the baldness genotype of little b, little b. Then we'll have the XY genotype with the dominant big B allele. And then the last one is the XY with the lowercase b allele. So looking at the phenotypes of these offspring, we have a 25% chance of getting a female offspring with a full head of hair, represented by this X big B, X little b genotype. So because the big B would mask the recessive allele here, this individual would show up with a full head of hair. There's also a 25% chance that these two individuals would create an offspring that is female with baldness. So this offspring here would get both little b recessive alleles. So meaning that because they have both recessive traits or recessive alleles here, this individual would show the recessive trait of baldness. Then there would be a 25% chance that the offspring would be a male with a full head of hair because they got that one dominant allele. And then lastly, a 25% chance of a male that is also bald. They have that recessive allele. Okay, so looking at these sex-linked traits, it's important to remember that the trait is represented as the exponent. You also need to include the X's and Y's to represent the sex of the individual, because these traits are specifically on those sex chromosomes.